Hi, everyone. We'll get started here in just a few minutes. Welcome back from lunch, everyone. This is the monitoring and research session. So this will be very similar to the morning. We'll go through each presentation. There'll be about 5 minutes at the end for Q&A. Please leave yourself on mute. If you want to leave your video up, that's fine. We like to see your faces. And if you have any questions throughout the presentation, please put them in the chat box. I did want to give a shout out for our workshops happening tomorrow. So we are starting earlier. We're starting at 9 a.m. tomorrow instead of 10, so pay attention to the times. We have a wildfire mitigation and restoration virtual field trip, followed by a climate smart approach to riparian restoration and revegetation. And then in the afternoon, you'll have a choice to pick between a mycorrhizal fungi workshop or a monitoring workshop. So the other thing I did want to mention is pay attention to the email that came out for tomorrow. There are 3 different Zoom links that you're going to need to use instead of just one. So with that, we'll go ahead and get started. So our first presenter is Bruce Orr with Stillwater Sciences. His presentation is titled Testing Methods to Improve Monitoring of Riparian Habitat Restoration Performance at Multiple Scales.
Good afternoon. My name is Bruce Orr. I'm from Stillwater Sciences, and I'm here today to talk about a pilot study my co-authors and other collaborators and I have been conducting to test various methods for improving monitoring of riparian habitat restoration performance at multiple scales. As always, this is a team effort. A lot of different people were involved, and the primary funding was provided by Valley Water. This pilot study is focused on the Guadalupe River in San Jose, California, in the heart of Silicon Valley. And since 1994, a variety of restoration and mitigation projects have been conducted on the river. And as part of the mitigation requirements, there are long-term adaptive management and monitoring requirements that need to be met with annual monitoring at a variety of these sites to test response in terms of riparian vegetation, recruitment, percent of native versus non-native cover, tree height and basal areas. But there are also a number of other aquatic and fisheries habitat-related monitoring components we won't be talking about today. I'll focus just on a subset of the riparian vegetation aspects. Here's a blow-up of the pilot study area. It's just a portion of the downtown project reach near downtown San Jose. And on the left here is a blow-up showing the transects labeled from 1 through 44. These are the transects used in the pilot study. For representative purposes today, I'll be focusing mainly on the lower transect number 44. As mentioned, the adaptive management program requires monitoring annually at a variety of sites. Vegetation components, there are short-term indicators such as plant survival, health, vigor, and recruitment. And there are longer-term indicators such as native versus non-native cover and riparian tree height and basal area. One of the challenges we faced after many years of doing fairly routine monitoring, in 2020, things changed. We had this combination of the worldwide pandemic, COVID-19, coupled with increased usage and distribution of human encampments along the riparian zone, coupled with threats from fire. Because of these safety hazards, over 25 to 30% of some of the monitoring sites were not able to be visited on the ground. This led us to initiate the pilot program to attempt to find alternative ways to gather data remotely that could be coupled with more limited field data collection to try to fill in the gaps and see if we could meet at least some of the requirements of long-term monitoring program. And an additional objective was to determine if using these remotely sensed data could help us extrapolate and expand from the site-specific transect data to more of a reach scale assessment of what's going on throughout the larger area. I'm sure all of you have heard a bit about LiDAR, or airborne laser mapping. We've talked a fair bit about it at this conference in recent years, including some of my prior talks. But typically in the past, we've talked mainly about use of LiDAR for generating a bare earth topographic surface or estimating canopy height. What I want to focus on today is more sophisticated use of LiDAR to assess three-dimensional structure of riparian vegetation. For the pilot study, we coupled field data collection along transects with the LiDAR data, and we used a line transect with circular plots established at regular intervals, basically a 10-foot radius circular plot established every 20 feet along the transect. And then the circular plot was extended vertically to create a cylindrical plot to get at a canopy density or canopy cover at different vertical layers. The understory, which is the less than 1 meter from the ground surface, the midstory, basically 1 to 3 meters, and the canopy, higher canopy layer, which is 3 meters and greater above the ground surface. We also looked at sort of total vegetative cover over the entire transect. And in the field, the amount of cover was differentiated between native and non-native species, but for the LiDAR transect analysis, we weren't able to differentiate species, let alone native versus non-native. Here's an example of some of the data on the left is a multispectral image, this case color infrared, just showing a portion of transect 44 and some of those circular plots that were established along the transect. On the right is a plot of LiDAR laser return data, color-coded to show relative elevation or height above the ground surface. So the blue dots are those laser returns from at or near the ground surface, whereas the red ones are higher in the canopy, in this case 10 to 30 feet above the ground surface. Here's a representative image. Essentially, this is a virtual belt transect taken along the line transect here. And the point cloud data just presented in this cross-sectional view to give you a quick general representation of the structure that we were seeing from the LiDAR. And then this was the basis for comparison with the field data collection. Transects ran from west to east. And here you can see I've highlighted the river channel towards the center of the cross-section. Here's zooming in on that river channel just to show a little more detail. In this case, the LiDAR vendor classified the points and color-coded. They're color-coded here. Points in blue are indicating water surface of the channel. Points in red are those classified as ground surface. Points in gray are sort of near ground cover or understory layer. And then points in green are the main vegetative canopy. Next, I just wanted to give you a field photo and a comparison with the LiDAR cross-section to give you a sense of an area where the field and the LiDAR comparison was working quite well. In this case, we have a relatively open riparian canopy, and at 30 feet point along the transect, the LiDAR data was doing a pretty good job of picking up the downwood and the racked tree limbs that you can see in the photo here. Should mention the photo and the cross-section don't align exactly, but the ovals give you a general area of comparison between the two. There was a sparse understory cover measured in the field at just 3%. You can see on the LiDAR image, there's really not much understory cover being picked up. Midstory cover was about 20%, which looks about right if you look at the LiDAR image. And then the taller canopy cover was 80%. So you can see we have, here's an example of pretty good correspondence between the two. To get a better statistical analysis and correlation across all transects, we prepared these plots, and our statistician did an analysis to look at the R-square values. And on the left, you can see here's the distribution of points. On the left axis, the y-axis is the LiDAR estimate of canopy cover. On the x-axis is the field estimate of canopy cover. And you can see for the overstory layer, which is basically half a foot to 3.28 feet or 0.15 to 1 meters, the LiDAR data tend to over-predict the amount of cover in this analysis. Mid-story layer, we also have a pretty low R-square. And in this example, you can see that the LiDAR is under-predicting the amount of mid-story canopy. And then over on the right, you can see the correlation between the LiDAR and the field estimates for the taller canopy cover at 3 meters and above. And there we have a good R-square, 0.74. And you can see on the plot that we have a pretty good correspondence. There's some scatter and noise, but LiDAR's doing a pretty good job of matching the field estimates. Just to go into this a little bit further, on the plot on the right, both these plots are looking at total canopy cover. The one on the left is looking at total canopy cover from 0.15 meters and above. Essentially, if you were laying on the ground looking up to the canopy, that's what you'd be seeing. R square is 0.52, not bad, but not great either. The LiDAR tends to over-predict to cover. And then on the right, you can see total canopy cover from 1 meters and above. And in this case, we've got a better correlation and a tighter fit. So this gives us a sense of the LiDAR is doing pretty well at the higher layers in the canopy, and we're having some challenges as we get closer to the ground surface. Here's just a summary of what we're picking up. And next here's just a reminder of where was the LiDAR not doing a good job of detecting conditions we were seeing on the ground. And in this case, I'll show you an example where it's the field observation showed a significant mid-story layer, whereas the LiDAR was not really picking that up well. Same with the backwater channel that was not picked up, apparently because it was covered by a dense willow thicket. And then we weren't able and didn't expect to be able to pick up and differentiate native versus non-native cover. Here's just that example I mentioned of the mid-story canopy. Look at the ovals. You can see the LiDAR is not showing much mid-story canopy, whereas on the left at the 50-foot station, field data suggested there was a 40% mid-story canopy. And then towards the left, between 250 to 280 feet, mid-story canopy estimated in the field was 70%. And as you can see from the LiDAR data, we're picking up almost no mid-story canopy. This suggests that the light penetration was being affected by the taller canopy and density of that canopy. Here's the case where the oval highlights the area where there's a dense willow thicket that apparently obscured and prevented the LiDAR from picking up a backwater channel. So what do we do about this? How do we improve? So for an overall canopy cover estimate, we're doing pretty good. But where we want finer vertical detail of the structure, we have some challenges. And what can we do about that? Well, one is trying to get higher density returns of LiDAR. Just maybe it was a situation where the LiDAR vendor could have done a better job, or maybe it met the technical specifications, but we need to increase what we're asking for. And that could be achieved also by increasing the overlap between the multiple passes of the plane and the LiDAR scanning, as well as mixing. Or right now we're focusing mainly on directly above orthogonal laser scans, but maybe if we had more oblique scans as well from each side of the riparian corridor, we'd start picking up a lot more of that lower vertical structure. And then another option is to use modeling to try to reconstruct the vertical structure based on our assumptions about the distribution of returns versus the distance from the top of the canopy towards the ground surface to account for that shading effect that might limit or block light penetration as the laser moves through the tree canopy and gets closer to the ground surface. And here's an example from prior studies that I've talked about at this conference, where we had high density research grade LiDAR collected by the National Center for Airborne Laser Mapping on the Santa Clara River in Southern California. In this case, they had 3 different lasers, each with a different wavelength, and gave us very high density returns. And this is an example where the LiDAR was doing quite a good job of picking up not only the upper canopy layers, but also all the way through down to the ground surface. Then here's another example from nearby where we have a different real canopy structure in the field. Basically 15-year-old willow tree recruits that all have a pretty even age stand. And you can see there appears to the LiDAR data are showing pretty dense canopy layer near the top, and then structure but less dense throughout. And then as you get near the ground surface, less vegetative structure. And that matches what we observed in the field. So we're doing a pretty good job there. So this is an indication that maybe there is something to do with just the quality or the density of the LiDAR, as well as perhaps the way they arrange the flight paths that might enable us to improve our results next time around. The other example of trying to improve our LiDAR results is shown here. In this case, we're rather than having just biologists try to interpret this, we work with a statistician and use the raw LiDAR data returns to then reconstruct a hypothesized distribution of canopy density that accounts for that potential blockage of shading by the upper layers. And this plot shows, one representation of that. You can see each line is at a different station, circular plot station along the transect, and where the black lines sort of jut out to the right there, we can see that that's an area with increased leaf density. So you get a sense from this plot of the vertical canopy structure. Here's another way of presenting those data. Basically to take that original LiDAR raw data returns and expand it using this hypothetical distribution of canopy density and multiple sampling then from that. And you can see it's giving us a little better picture of what that canopy structure might look like. And here's a more direct comparison. If you look at the bar on the right, that's from the farthest station to the east on the transect, and the arrow shows it's linked to the LiDAR data. The LiDAR picks up that taller canopy quite well, but as you can see, as you go down through to the mid layer and below, the hypothetical distribution on the top indicates a greater leaf density than what we're actually seeing on the raw LiDAR returns. So to summarize, our standard LiDAR approach worked well for estimating canopy height, estimating total canopy cover, and estimating cover in the tree layer itself. We can also use standard LiDAR for capturing 3D vegetation structure in the more open areas, but we definitely are having problems in the denser areas of riparian forest. And as I mentioned, we can also use the LiDAR to generate a bare earth topographic surface like we've been doing for years, and that could serve as a high-resolution base elevation that could be coupled with subsequent drone or UAV photography to track canopy height changes through time without having to repeat LiDAR at frequent intervals. And then on the right-hand side, we have a couple possible solutions to the problems or challenges we faced. As I mentioned, one is trying to develop custom technical specifications for higher density and maybe multiple swath and multiple angle LiDAR to solve some of those problem areas. And the other is, which we're beginning to explore, is combining the LiDAR with multi or hyperspectral imagery to see if we can start teasing out differentiating native versus non-native vegetation, or ideally the holy grail would be to differentiate individual species. And with that, I'd just like to acknowledge the funding from Valley Water again, and the technical assistance from co-authors, and then Peter Baker, Aaron Elsey, Ian Pryor, and Carly Rodriguez at Stillwater Sciences. And I'd be happy to answer questions now. Thank you.
Thank you, Bruce. Great presentation. We have a few questions that I think one of them you already probably touched on, but the slide of what LiDAR did detect went by a little fast. Hope you can return to that at the end, which you did a great job of summarizing that at the end, but I didn't know if you wanted to elaborate that on anymore.
Yeah, hopefully that summary covers it pretty well. this pilot study of the Guadalupe River, I was underwhelmed by the quality of the LiDAR compared to what we had seen on some other projects, like that example at the end from the Santa Clara River. So these results are hot off the presses, so we just got this in the last couple of days. So we're still sort of digesting it all and what it all means. So one of the areas I really want to do is look into whether or not the LiDAR that was provided met the technical specifications we had set up. In this case, we had told our client what we wanted, but they contracted directly with the LiDAR vendor. So we were a bit out of that final loop, and maybe something got lost in the translation there. So I think depending on what the pilot project, we're doing a great job in picking sort of total canopy cover from like 1 meter and above. I think the correlation there is quite good, as well as the simpler metric of canopy height is being picked up quite well. So those upper layers, we could also do some finer stratification. It wasn't part of this project, but in other studies, we're looking at like 1 meter vertical bands. And with the higher resolution LiDAR data, like we have on the Santa Clara River, we're actually able to differentiate those pretty much from the ground surface on up, although there's still a few areas where there appear to be cases where there is potential blockage by that upper canopy, and we're not getting a fully accurate representation of the lower layers. And we're exploring different ways to test that. One of the challenges, I think, of using this method for repeat time series sampling over a number of years is the technology changes, sensors change. It's sort of like Pam was talking about earlier in Landsat imagery, you've got 3 different sets of sensors you've got to try to calibrate. So they're different. That's where working with the statistician and talking to other researchers about how they're approaching that, I think will really help us. So LiDAR's got great potential. Should have mentioned this was all done with standard piloted aircraft LiDAR. In future studies, we hope to start looking at drone LiDAR as well, and just comparing those cases. And perhaps the ideal study, we'd also have a ground-based LiDAR to pick up that detailed lower layer canopy structure and compare it with the LiDAR data taken from above. So
Great, thank you.
That fully answers the question, but I'm happy to talk later with anyone, provide more details.
Sure, that's great. And I'll also remind everyone too that the presentation is recorded too, and we'll be sending that link out, so you can always go back and look through any of the slides. You answered the question already on whether it was drone mounted or not. Is there a current threshold at which the cost and computational time of increasing LiDAR resolution, processing, modeling, analysis, etc., exceeds the cost of humans collecting data on the ground?
That's a very good question. Yeah, I think there's definitely a scale factor. So if you're doing a small project and you've got readily available local labor, then it's probably sticking with tried and true methods makes a lot of sense. If you're talking like this project, the pilot study was focused on a fairly short sub-reach with a half dozen or so transects that were the main focus. But if you really want to be looking at larger reach scale efforts or full river corridors of 50 to 100 miles, then combining the LiDAR with some focused ground truthing and field data collection seems like the best way to go to really track changes in these systems over time. So it's similar to what I've talked about in the past, how we use past talks, how we use GIS to do our eco-hydrological assessment to plan restoration implementation, and then the same approach, like coupling for the larger scale projects, coupling the remote sensing and GIS analysis with the field data is really the way to go. If I have my druthers, we've got 1 or 2 other pilot projects we're trying to get underway or secure funding for that would dive into some of these issues in more detail. And hopefully at least one of those we can address some of the cost efficiency issues more directly.
Great. Thank you very much for your time. We really appreciate you participating in our conference again.
Thank you all for listening.
All right, so up next we have Annie Henry with the University of Denver. Presentation title is Cover of Tamarisk Covaries with Regional and Local Environmental Factors to explain the functional composition of riparian plant communities.
Hi, my name is Annie Henry, and I'm a graduate student at the University of Denver. Today I'm going to be presenting some of my dissertation research to you. This project is entitled Cover of an Invasive Tree Co-Varies with Environmental Factors to Explain the Functional Composition of Riparian Plant Communities. In general, my research deals with this big picture question: what happens to plant communities when tamarix is defoliated by the biocontrol beetle Diorama? So far, studies have shown a wide range of plant community responses to defoliation, including no change in understory plant community, small increases in native vegetation and diversity, shifts towards more of an upland community rather than the typical riparian species, and increased secondary invasions. So what causes such a wide range of responses? In other words, what are the mechanisms underlying plant community response to tamarix cover? To answer this question, we're looking at plant community response using traits rather than species identity. Looking at traits makes sense given that a plant's traits, in large part, are what determine its ability to thrive in a given environment. So traits are the direct link between abiotic conditions and plant community structure. Environmental change, such as flow regulation, invasive species establishment, or invasive species removal, changes the environmental filters operating in a community. So it changes the environment. And this in turn changes the traits that are advantageous in that plant community, finally resulting in a community made up of different species. So looking at traits rather than species sheds light on what abiotic conditions are really driving the community. Why does this matter? Well, this is helpful for anticipating plant community response to invasive species establishment and removal, including anticipating the traits of secondary invaders, and then selecting appropriate species for revegetation. Ultimately, the plant community contributes to ecosystem functions such as habitat provisioning and nutrient cycling, so it's really important that we understand what's going on. This study looks at a snapshot of tamarix cover across a large geographic area and asks, what traits are most important in defining riparian plant guilds? And by guilds, I mean groups of species with similar traits. Secondly, how does guild cover vary along a gradient of tamarix cover and abiotic condition? And lastly, how does guild diversity respond to tamarix cover and abiotic condition? So we selected 95 sites ranging from low tamarix cover, more native-dominated plant communities, to plant communities with overstories made up entirely of tamarix. Sites were along the Green River, Colorado River, and the Dolores River, and tributaries connected to those. And all of these sites included in this study had exposure to biocontrol beetle between 5 to 9 years. So these were all part of a larger study published in 2017, looking at the response of vegetation to tamarix removal across the southwestern United States. For our purposes, we examined all sites in that larger study that only had biological control removal of tamarix. And vegetation data was compiled by Gonzalez and others and was sampled between 2012 and 2014 using line intercept method. For this study, we compiled data on 17 traits for 139 species present in our sites using online databases and literature searches. This includes true biological traits that primarily relate to resource acquisition and reproduction, as well as what might more accurately be called ecological affinities, so ordinal values that give an overall measure of the plant's response to things like drought, flood, etc. So we chose these traits based on previous studies that have shown them to be relevant in defining riparian plant communities. We also collected data on abiotic factors, elevation, precipitation, river width, distance to river water's edge, soil electroconductivity, and elevation above the water channel to try and parse out the influence of tamarix cover and these factors. So question one, what traits are most important in defining riparian plant guilds? So the first step in this process is just identifying and defining the plant guilds that are present across the study area. To do this, we used a cluster analysis that grouped species with similar traits. And to visualize them, we plotted the guilds as well as the weighted averages of all the traits in a principal coordinate analysis, or PCOA. And a PCOA is simply a way of ordering multivariate data into 2 dimensions so you can see relationships among different variables. So what you see here is the first 2 axes of the PCOA. Each point is a species and the symbol is its guild identity, and we'll get into the guilds more in a minute. The text represents the weighted averages or centroids of trait values. So the farther out on an axis a trait is, the stronger it is in defining that axis. And I only plotted the top traits here, so there are a whole bunch of traits that were also just clustered around the center that aren't important in defining the plant community. The blue text represents variables that were not included in the analysis, but we calculated the centroids after the fact to help interpret the results. The main result here is that reproductive traits, such as the ability to reproduce asexually, represented here by clonal, seed weight, and perennation, so perennial versus annual, are major factors in defining plant guilds. As are resource acquisition traits like life form, height, and specific leaf area. That's what this stands for. Our cluster analysis identified 9 guilds defined by differences in those traits. And here I'm just going to talk about the 7 understory guilds. A primary distinction in these guilds was the ability to reproduce asexually. So we have non-clonal guilds that can't reproduce asexually and clonal guilds that can. These are radial plots that just show a summary of average trait values for each guild. So the further out on this little spiderweb line is, or point is, the higher that trait value is for that guild. So the non-clonal guilds all tended to have moderate to long bloom periods and light seeds. They were further divided by annuals and perennials. The clonal annuals were further divided by graminoids and forbs. So we have annual graminoids, which were anaerobic tolerant, for example, barnyard grass, and then annual forbs that were drought tolerant, for example, common kochia. Now on the perennial side of things, these 2 guilds were differentiated primarily by specific leaf area. So we had a guild of high specific leaf area species. These are the resource acquisitive species. For example, narrow leaf plantain. And then a resource conservative guild with a much lower specific leaf area. And broom snakeweed is an example species in this guild. There were 3 different clonal guilds. First, we have drought tolerant perennials, such as Canada thistle, anaerobic tolerant perennials, for example, common reed, and lastly, a resource conservative guild with super low SLA, for example, greasewood, so more upland species. So question 1, what traits are most important in defining riparian plant guilds? First, reproductive traits, which tend to be strongly linked to disturbance, such as flooding and fire. This suggests a gradient of disturbances present throughout the study area. Second, height, specific leaf area, and life form were important, suggesting that there's also a wide range of drought versus waterlogging tolerance, as well as resource acquisitive versus conservative strategies that are successful throughout the study area. So my second question, how does guild cover vary along a gradient of Tamarack's cover and abiotic condition? To answer this question, we used a redundancy analysis, or RDA. This is essentially like a regression analysis, but with multiple explanatory and response variables. So the position of an explanatory variable along this axis is what shows what defines this axis. And then the position of the guilds shows what sites have a higher cover of those guilds. Together, this shows the relationship between gilt cover and the environment. In this figure, I'm starting out with just the environmental factors that define these axes. So you can ignore these little red numbers. And the length of an arrow is relative to the importance of that variable. So at high levels of RDA1 here, we have sites that are far from water, have high Tamarix cover, both absolute and relative, and high elevation above water level. So these sites are far away from the elevation and precipitation, so that means they have lower elevation above sea level and lower precipitation. So these are our dry Tamarix dominated sites. Okay, so I should mention here that elevation above sea level is also negatively correlated with temperature. So over here, the low sites are also hotter. Now on the other side of the RDA, this RDA1, we have the opposite types of sites. So high elevation above sea level, higher precipitation, closer to water, and low Tamarix cover. Now I'll show you where the gilts are on this figure. Okay, so the gilt points on the RDA are showing you where the sites with high cover of each of these gilts are positioned on these axes. So here we're looking at non-clonal gilts, so only sexually reproducing, and you can see that 3 out of 4 of these gilts are associated with sites far from water and higher Tamarix cover. So these 3 over here, these 3. We were able to do a follow-up analysis with mixed effects models, and we were able to show that annual forbs increased significantly with Tamarix cover. So there's some stats for you for that. So this gilt actually has the highest number of invasive understory species also. These tend to have an annual life cycle, reproduce with many small seeds, and have a moderate to long bloom period. Additionally, these species tend to have a higher specific leaf area than one would expect in a dry, resource-poor environment. So this suggests shade tolerance. So these are the types of traits we find in more Tamarix-dominated sites. On the other side of things, we have the clonal anaerobic tolerant perennials, which were, we were able to do a follow-up analysis with that and found that they were significantly correlated with both Tamarix cover and distance to water. So these anaerobic tolerant perennials are more common in wetter sites with lower Tamarix cover. And then the clonal drought tolerant perennials were related to distance to water here, but not strongly related to Tamarix cover. So this suggests that as Tamarix is removed and sites become hotter and drier, this gilt may increase, which incidentally is also made up of a fair number of invasive species. They just tend to be different kinds. They are longer lived, more drought tolerant, and can reproduce asexually, excuse me. So for the whole RDA that I just showed you, we explained 31.5% of the variability in gilt cover. And this is just to show you that almost 9% is just explained by Tamarix alone. So while Tamarix does co-vary with environmental gradients, it also has an influence on the plant community separate from the environment. So how does gilt cover vary along these gradients? First, Tamarix cover favors a short life cycle, so annuals, with lots of seed production and shade tolerance. Drought-tolerant perennials are likely to replace annuals as Tamarix is removed. And the typical riparian gilt clonal anaerobic tolerant species are negatively affected by Tamarix. So while Tamarix does co-vary with environmental variables, it creates a filter independent of other environmental factors. So last question real quick, how does gilt diversity respond to Tamarix cover in abiotic condition? So this figure shows that axis of RDA1 from before, but plotted against gilt diversity, measured as Shannon diversity's index for gilt cover. So what we found was that mid-ranges of both tamarix cover and environmental conditions are associated with higher gill diversity. So in here we have the highest gill diversity. So very dry as well as very wet sites have a much lower diversity, and the same is true of tamarix cover. Very high tamarix cover and very low tamarix cover have lower diversity than intermediate levels. So this relationship was confirmed with a quadratic mixed effects model, and the RDA1 explains 43% of the variation in gill diversity. So how does gill diversity respond to tamarix and abiotic condition? Basically, plant communities are more specialized at both low and high levels of tamarix, as well as environmental extremes. Okay, so the take-home messages of this study are first, that we've highlighted this role of tamarix as both a passenger and a driver of plant community function. Tamarix facilitates non-native species because of their drought tolerance and shade tolerance and their high reproductive output. As tamarix is defoliated, understory plant communities may shift more towards those clonal drought-tolerant guilds. And lastly, native species from that guild could be chosen for revegetation in order to compete with non-native secondary invaders. So it looks like I probably have a few minutes for questions. Just want to acknowledge the funding sources and people who have helped out on this study, and I'll take some questions. Thank you.
Thanks, Annie.
No problem.
Before we have any questions come in, I just wanted to wish you good luck. Annie has her dissertation tomorrow, so I just wanted to, yeah, you're welcome. If you do have any questions, please put them in the chat box. We are running a little bit behind, so we might just keep it rolling, but I did want to just make one comment. Your take-home messages, I think, are so important for messaging as we go out and do riparian restoration, right? And that what to do before and after. And you had some really, really great points in there. So
great. Thank you.
Yeah, thank you. It doesn't look like we have any other questions come in, so I think we'll go ahead and move on to the next presentation. Annie, thank you for your time. And again, good luck tomorrow.
Thanks.
Okay, get us back on track here. So the next presenter is Kristen Dillon with the Bureau of Reclamation. And presentation title, The Impacts of the Tamarix Beetle on Southwestern Willow Flycatcher Habitat on the Middle Rio Grande in New Mexico.
One of the largest remaining southwestern willow flycatcher populations breeds along the Rio Grande in New Mexico. Back in 2015, we had seen the impacts of Diorama on swivel breeding habitat in other parts of the Southwest, and we're starting to hear reports of the beetle on the Rio Grande, but hadn't yet seen it in occupied swivel habitat. But we knew it was only a matter of time. So that year, 2015, we began a Diorama impact monitoring study in occupied swivel habitat on the Middle Rio. Sure enough, in 2016, Diorama was observed throughout the Rio Grande, including at some of our monitoring sites. This is the map I'm sure everyone here has seen that RiversEdge West puts together every year, showing Diorama locations by year. This map includes 2007 to 2019 observations, with the 2019 Diorama locations in blue. We work here on the Rio Grande from approximately Albuquerque to El Paso, and Diorama had colonized this entire area by 2016. So it seemed important to initiate this study because Diorama is likely to have a significant negative impact on swivel habitat in the Rio Grande, where, like most modern day swivel habitat, salt cedar is a major component. Although the Rio Grande floodplain was historically native dominated due to changes in hydrology and vegetation composition, salt cedar has overtaken willow in its dominance of the riparian ecosystem. This graph shows the percentage of nests constructed in salt cedar versus willow over the last 2 decades. And as you can see, it's shifted from approximately 75% of nests constructed in willow back in 2002 to about 2/3 to 3/4 of nests constructed in salt cedar currently. Likewise, about 75% of breeding territories are either salt cedar dominated or have a salt cedar component. So we're using photographic monitoring to quantify beetle-induced changes in canopy cover and vegetation community structure in Swiffle territories. So hemispherical photos are taken from within the riparian vegetation, facing upwards at the vegetation canopy using a fisheye lens. And landscape photos are taken with a normal lens facing inwards towards the vegetation community. And then we use a software program that's designed to classify the hemispherical photos and calculate canopy cover. 2015 and 2016 provided pre-impact, pre-Diorabda defoliation data in most of our sites. We did not observe Diorabda in our monitoring sites until 2017. So typically, as is likely intuitive to everybody here, canopy cover increases throughout the growing season. So this graph shows the seasonal progression of canopy cover in our native dominated control site as measured by hemispherical photography. So we take photos 3 times per breeding season in May, June, and July. And as would be expected, canopy cover increases throughout the summer as vegetation leaves out. However, we see the opposite trend in sites impacted by Diorabda. So this graph shows canopy cover as measured by hemispherical photography, and one of our monitoring sites has been hit hardest by the Tamarisk beetle, DL12 North. And I'm going to continue to use this site as the example throughout this talk. So as you can see, canopy cover declines throughout the breeding season as defoliation occurs. In May of 2017, there had been no defoliation in this site yet. In that first year of defoliation, canopy cover starts around 92%, and by July of that year, declines to about 85%. In the second year of defoliation in that site, 2018, average canopy cover for the site starts a little bit lower, just under 90%, and continues declining even further to just over 80% on average by the end of the season. Now, Diorabda did not colonize this site in 2019 or 2020, giving the habitat a chance to recover. However, despite 2 growing seasons without defoliation, the site still has not recovered to pre-Diorabda canopy density. And instead, canopy cover seems to hover around 85 to 90%. So this line shows average canopy cover for the site prior to any defoliation. And again, it was over 90%. And despite 2 years without defoliation events, the site has not returned to that level of canopy cover. So we'll come back to why this might be later. So now I'm going to show you what the landscape photography looks like. And these photographs really give an excellent depiction of what that defoliation-induced decline in canopy cover that we just saw graphed really looks like in the habitat. So this first photo is DL12 North, same site I just showed you the graphs for, in May of 2017, prior to any Diorabda defoliation. By July of 2017, this is that same location. So these are photographs taken from a fixed point. And by July of 2018, after a second year of defoliation, this is how the habitat looked. So I want to point out here that hemispherical photography cannot distinguish between live and dead vegetation. As you can see in these second 2 photographs, there's still quite a bit of dead foliage on the salt cedar. But the difference between live and dead foliage is likely significant to a nesting bird. So when hemispherical photography indicates 80% canopy cover, that habitat can actually look quite poor despite what sounds like a fairly high cover percentage. So remember, those previous graphs, hemispherical photos showed us that average canopy cover of the site was 92% in May of 2017 and declined to about 80% by July of 2018. But clearly, this looks like a much larger than 10% decline when you actually look at it. And then finally, this last photograph shows the same location in July of 2020, after 2 seasons with no defoliation. Clearly, there's been significant habitat recovery. But if you look closely, as we see with the hemispherical photography, it's still not returned to the pre-defoliation levels. So if you compare those first and last photos in the series, you can see, for example, in July of 2020, there's much more sky visible. You can see those exposed tree trunks in the last photo. So clearly the vegetation has not recovered to the same density that we saw in May of 2017. In addition to taking hemispherical photos throughout the breeding season, we also took photos once per winter as a means of measuring woody mass and determining what percentage of breeding season cover was foliage. So when we took these photos in the winter, there was no foliage on the trees. This is just cover due to branches, tree trunks, woody mass alone. This graph shows average dormant season canopy cover in DL12 North, our same focal site here. And there's a few things of interest that I want to point out. One is that a high percentage of cover in these sites is comprised of woody mass alone, right? So if you look at the 2017 photos on this graph, a little over 80% on average canopy cover is measured in the winter. So that means when we see 92% cover in the summer, you can kind of do some back of the napkin math and figure out how much of that is foliage. But when we see breeding season cover drop to nearly 80%, that translates to very little foliage left on the trees. The other thing to notice here is the obvious decline in woody mass over time. So again, this graph is not within the breeding season. This is dormant season cover across years. Diorama is expected to slowly kill salt cedar through the stress of repeated defoliation, which leads to the loss of woody and root mass. And that's exactly what we're seeing here. Particularly after the second year of defoliation in 2018, we see this big drop in woody mass. And this can, again, be compared to our control site, less than a kilometer south, where woody mass essentially stays the same over time. So this control site is predominantly coyote willow. So we had a wet year in 2019. We see this slight increase in woody mass, and then it kind of declines a little bit. But essentially, woody mass in this site stays more or less the same over time, which is what we'd expect, compared to this steep drop-off in DL12 North. So I said earlier we would come back to the fact that although it's been 2 years without defoliation in these monitoring sites, the vegetation still not returned to pre-Diorama health, as we saw through hemispherical and landscape photos. It seems like there's likely a few things going on here. First of all, we saw that the stress of defoliation led to declines in woody mass, and this contributes to an overall reduction in canopy cover. However, just looking at these 2 photos, one of May in 2017, one in May of 2020, you can see there's an overall decline in foliage density as well. Additionally, we've observed declines in canopy cover in some of our monitoring sites that were not impacted by Diorama. So it seems likely the vegetation is simultaneously being stressed by drought and a dropping water table, as well as Diorama. So in some sites that aren't impacted by Diorama, drought alone is enough to cause decline in vegetation health. But there appears to be an interactive effect of drought and defoliation in some sites that's causing these severe declines in vegetation health and a lack of recovery even in the absence of defoliation events. So in addition to photographic monitoring, in 2018, we began quantifying vegetation composition and density at our monitoring stations in 3 different height class in order to track potential longer-term changes in the vegetation community. Just very briefly, although we only have 3 years of data at this point, our initial results suggest that there are increases in dead vegetation at the sites that have been impacted by Diorama that seem to correspond with nearly identical decreases in salt cedar. So the dark bars on this graph are showing the increases in dead vegetation over time. The lighter bars are showing decreases in salt cedar. We do not see a corresponding increase in other types of woody vegetation, such as coyote willow. We're just seeing this increase in dead vegetation. So these changes in canopy cover and vegetation composition we've been discussing can affect breeding flycatchers both directly through loss of nest concealment, as well as indirectly through changes in microclimate. So we deployed data loggers to measure temperature and humidity at our hemispherical photo stations. Each line indicates one photo station within a single site. So each of these lines here is within a single site. This is our control site. We see little variation in maximum temperature between photo stations within a site at our control site. So native dominated vegetation here. And maximum temperatures of approximately 85 to 95 degrees. However, at defoliated site, we see a noticeable difference in maximum temperature between photo stations within a site. And keep in mind, these photo stations are only about 50 to 75 meters apart. So the difference between points lies in the percentage of the vegetation at that point that's comprised of salt cedar compared to other species. So those points depicted by the lower two lines in the graph had minimal salt cedar and therefore also minimal defoliation. And maximum temperatures remain low, similar to the 85 to 95 degree range seen in the control site. However, the 3 points depicted by the upper 3 lines on the graph, the blue, green, and purple, were dominated by salt cedar, nearly 100% in some points. These locations were severely defoliated by Diorama, and maximum temperatures here were much warmer than at the other 2 points in the site, often over 100 degrees. So again, these differences are likely very important to a bird trying to regulate their own temperature, as well as the temperature of their eggs and offspring. We saw similar patterns in minimum relative humidity, such that defoliated sites were more arid. In other words, despite a relatively small decline in canopy covered, only about 10% or so, there's a measurable increase in temperature and humidity. So a small loss of canopy cover can have a severe impact on breeding willow flycatchers, both directly through the loss of nest concealment and indirectly through a loss of suitable microclimate conditions for nesting. And clearly you can just see the difference in this front photo where the nest circled here in red is in fully foliated salt cedar compared to the other two where the salt cedar they're nesting in is being defoliated. So of course the big question here is, how is this affecting the birds? And this graph shows the number of nests and nest productivity in Dale 12 North over the last several years. Although we need additional years of data to really determine a long-term trend, the data we do have suggests a slow overall decline in both nest numbers and productivity in this site. And even just anecdotally, the habitat in the site does appear to be declining in quality. However, we haven't seen as steep a decline in habitat conditions in all of our monitoring sites that have been impacted by Diorama. The take-home message here is that Diorama certainly negatively impacts habitat quality for willow flycatchers during defoliation events. And the stress to vegetation can be severe enough to prevent immediate recovery in the absence of Diorama. In addition, those impacts can be interactive when combined with other environmental stressors, such as drought, making the impacts of Diorama more severe and longer lasting than what we might see due to defoliation alone, as well as uneven across breeding areas as environmental conditions might vary. So in many cases, not only does it appear highly unlikely that other non-salt cedar woody species will quickly, or in some cases ever, take over the space opened by dying salt cedar, our data suggests that simply removing Diorama from the impacted habitat might not be enough to facilitate the recovery of the habitat and the vegetation that's already there to its pre-Diorama state. And so with that, I can take any questions if I have any time left.
Thank you, Kristen.
Thanks.
Great presentation. We do have one question that came in. I wonder about ground-based LiDAR pointed upwards for ground truthing or at least cross-checking.
Yeah, that's a great suggestion. It's not something that we've tried yet, but I think that would be a great way to kind of compare measurements of canopy cover and see if they differ between these different measuring methods. Yeah, I think that'd be a great path to take moving forward, but it's not something we've done yet.
Okay. There is a comment from Bruce Orr that I do just want to read out loud. I'm happy to see the use of hemispherical photos to track canopy cover. We've used that method in various studies of riparian shading for stream temperature modeling, and I'd like to use that for our next test of LiDAR estimation of vertical canopy cover. So, and then it looks like someone had a question about the software program that analyzes the hemispheric photos, and David Moore chimed in, we use HemiView.
Yeah, Dave and I work together on this project. HemiView is the software that we use to analyze the photos and classify them into cover versus open sky.
Great. And then, of course, Marianne asked if you're going to publish this, and that it corroborates everything we've observed on the Lower Colorado River. So very interesting. I think especially with the changing climate and the decrease in water overall is going to be interesting with what happens with a lot of these areas that have already been stressed by the beetle. So.
Definitely.
All right. Well, thank you very much for your time. We really appreciate it.
Thank you.
Mm-hmm. Okay, up next, we have Andrea Hummel and Joe Schroeder with Tetra Tech. Presentation title, Preparing the Southwestern Willow Flycatcher for the Tamarisk Leaf Beetle in the Middle Rio Grande, New Mexico.
I'll do a quick introduction here. I know many of you, but I know we have a lot of new faces that I hope to meet and greet someday here soon. I'm a senior environmental scientist with Tetra Tech, and I've been working on the Middle Rio Grande throughout the West and Southwest for the past 20 plus years with many of you, and I know a lot of us are saying 20 plus years, so that's the term I'm sticking with from here on out. Doing a lot of restoration work, restoration on the river in and around Albuquerque, all the associated, compliance that goes along with that. Previous to Tetra Tech, I was with the Corps of Engineers for almost 14 years, and City of Albuquerque Open Space prior to that. So I've been doing a lot of work related to the species and the collaborative program. Joe joined Tetra Tech about 3 years ago, and he's done a lot of work throughout the West and Southwest on wetland delineation, permitting, also wetland restoration, upland restoration. He's a great botanist in terms of habitat sampling and mapping, and then especially the remote sensing that Chris talked about, LiDAR, GIS modeling, and he's going to be talking about that and some of the results for this study. So I just wanted to give a little bit of background to this. There was actually a first part to this study that was published in 2015. We worked on from 2013 to 2015. When I was with the Corps of Engineers, I was the project manager, and we had Tetra Tech do the work for the collaborative program. And at that point in time, as Chris had mentioned, we were starting to see a lot more Tamarisk leaf beetle activity, and that impacting southwestern willow flycatcher habitat. And what could we as the program do in terms of trying to get ahead in terms of restoration planning? So Tetra Tech at that time pulled together all the information on, what we had in terms of plant information, Hinkenomart, territory data, fire, salt cedar, Tamarisk leaf beetle, and other stressors. And initially in that 2015 report, identified 25 areas with 103 potential sites. So if you look at the map here to the right, that's an area, and then there's a number of sites, and their different colors are different priority levels. At that time, the study went from Los Lunas down to the Elephant Butte Reservoir. So in 2018-19, the Corps and the program asked us to then update that study, both knowing we had further effects of Tamarus leaf beetle, which I'll show you some graphics here in a minute, but also evaluating the changes since the original study, trying to look a little more at the metapopulation connection. So we had a lot of territories, smaller territories in different areas we didn't have them before. How could we connect them more so that the birds could move throughout the system? And then again, still trying to get ahead in terms of restoration that could be done prior to additional Tamarus leaf beetle impacts. So the current study contained all of these components. I'll just show you real quick and I'll come back to that. So Tamarus leaf beetle through 2018, as you can see here, we knew we were going to be getting to the point where it was converging from north and south in the Mid-Rio Grande into the Albuquerque Socorro area, so that by 2019 you can see the straight line through there. So in that effect, Tamarus leaf beetle is basically throughout the Middle Rio Grande. So it was sort of a considered, we didn't have to necessarily map it. We knew it was there everywhere. Also, the current study updated the vegetation. Originally, we used the 2012 Hinkino-Mart mapping. We updated to the 2016 Hinkino-Mart mapping. And then we also looked specifically at these sort of A and B groups. What are sites that have currently greater than 25% salt cedar, but also sites that have less than 25% salt cedar? Maybe they would be a little easier to do some restoration work on. Also updated the groundwater model, had since the previous model more data that could be included there to make it a little stronger. Also looked more specifically and coordinated with Fish and Wildlife Service on, let's look at the territories and trying to get that distance between 10 miles is the number we were getting so that they could have places to go in between. And then also looking at areas where they did not want restoration to occur because the birds were doing well there. And then also some particular areas they wanted us to look at. Also, since that time, a lot more completed habitat restoration projects and unfortunately fires. And then within all that, there were some anomalies. So we did look at a couple other different things just to verify. We looked at the habitat suitability model, which also used the 2016 Hinkino-Mart data. Also the USGS habitat model, which was further verification when looking at sites to verify what was going on there. Some normalized difference vegetation index verification. And throughout all that, also came up with some alternative sites. So now Joe is going to talk about the results.
Great, thank you, Andrea. That was a great overview for the project. I'm going to take the remainder of our presentation time walking through the process that we went through to develop the flycatcher Tamarus leaf beetle habitat restoration siting model. So we started the project with a deep dive into the flycatcher nesting habitat literature and spatial data sets available for the Middle Rio Grande. Once that was completed, we set up several meetings with the collaborative joint work groups, which included subject matter experts, researchers, practitioners, regulators, and land managers. Frankly, most of the participants in those meetings wore most of those hats. But during those meetings, we presented our current findings and communicated the direction that we were taking the study, and then we facilitated an open forum to discuss the opportunities and constraints, and to pretty much make sure everybody was moving in the same direction, and we all agreed on what direction that was. Which can be challenging, but it was they were all very productive meetings. One of the more obvious benefits that resulted from the early collaboration was a clear definition of our area of interest. It was honed down to between Los Lunas and the full pool extent of Elephant Butte Reservoir. So aside from just getting the final study area finalized, that was really just the tip of the iceberg in terms of the value that the meetings yielded. It's hard to overstate the value of getting a bunch of smart people in a room and talking about and linking their respective specialties towards a common goal. So Big thanks to everybody in the program that helped us and helped us make those meetings successful. Let's go to the next slide. Once the study area was defined, we moved into the beginning stages of the site selection process. Now, the Hink and Omar community structure vegetation mapping was an extremely powerful tool for the project, and we used it as the primary classifier for the analysis. Once that was, established, we then looked at what we were considering hard constraints, which included things like a patch size. We wanted a patch to be greater than 5 acres and or be expandable or link into adjacent habitat to have it a minimum 5-acre patch size. The next hard filter was a depth of water. And so once, because obviously the habitat creation for flycatcher is going to require depth of water that is suitable for the coyote willow and tree willow to become established and persist. So once we got those hard filters in place in the model, we ended up with approximately 200 potential restoration sites within the study area. We enumerated those sites and then began looking at the secondary filters, such as ease of transition from unsuitable to suitable habitat. Again, going back to the Hink and Omar community structure mapping within the project area. And we looked at ecological site potential, which we interpreted via the normalized difference vegetation indexing, and using multiple years of aerial imagery. We also looked at the inundation mapping, LiDAR data were available, and a long list of other spatial data sets. This review allowed us to identify 50 sites with the highest restoration potential for the flycatcher. Once we had our 50 semifinal sites, we initiated field activities to field validate the sites. Field validation included depth of water evaluation, soils, indicators of inundation, vegetation type validation, vegetation density, as well as access. I think access and land ownership are two really important components of our restoration project, because we wanted to make sure we're providing practical sites, and that includes sites that added value, which includes easy access and constructability. We can go to the next slide. So through the field validation, we honed the 50 semifinal sites to 9 final high priority sites. The sites represent the low-hanging fruit in terms of flycatcher restoration. It's based on some, value items such as ease of access, cost effectiveness, metapopulation, habitat resiliency value. It's kind of a mouthful, and as well as ownership, as I mentioned before. Now, just because we came up with a list of the 9 final sites doesn't mean that the 50 semi-final sites don't offer value. So we made sure to memorialize a master habitat restoration siting matrix for all 50 sites within the study's report. Let's go to the next slide. Okay, so this is site 1. Also known as A43, based on the original enumeration for the sites within the project. And this is the type of information that we compiled and present in the final flycatcher report, or flycatcher habitat restoration siting report. I think we are a little bit short on time, so I'll just go through some of the outline of it. So we're looking at the site size, post-restoration habitat patch size. It was a really big one, so we want to create large habitat patches that allow for large populations. And we also get into the vegetation habitat hydrology disturbance history. Yellow-billed cuckoo is something I want to make sure we bring that up, because that was a complicated hurdle to overcome for the project, and we had to eliminate several sites because they were nearby yellow-billed cuckoo habitat or corresponded to yellow-billed cuckoo habitat, and bad practice to take habitat from one endangered species to create habitat for another one. All right, we could probably go to the next slide. Okay, so then we get into the lessons learned, and this was a challenge. How do you come up with a lessons learned slide for a study that took 2 years and consumed a lot of your bandwidth for those 2 years? But at the end of the day, I guess some of the high level lessons learned on this project and some of the value that we were able to glean from the project was just a full spatial data inventory for the Middle Rio Grande. We looked at every single piece of data that we could get our hands on and compared it to flycatcher nesting, both present and past, to see which ones correlated to flycatcher nestings, and then also basically looked at the value of each one of those data sets in terms of precision and accuracy, and just went through our own filtering process for all of the data sets within the Middle Rio Grande. And so some of the really big ticket items in terms of the value presented to the project included the inundation mapping. I can't say enough about this. Everybody knows about it. I think probably more so than any other data set, the mapped inundation from previous floods in the Middle Rio Grande corresponded to flycatcher nesting more than anything else. Also looking at the streamflow modeling, which allowed us to look at the potential for inundation in any given year along the entire Rio Grande or Middle Rio Grande. NDVI was priceless. Vegetation density with LiDAR was, especially when paired with NDVI, where we get vegetation height and density and productivity. I mean, that was a very powerful tool when we're looking at the probability of flycatchers utilizing an area, or even in this case, evaluating site potential. Because it stands to reason that sites that have very high density of mixed tamarisk, salt cedar, and native species is going to support high density native species, which obviously the flycatcher require. And then vegetation type and patch size, again, with the going back to the Hank and Omar, that's been a long-standing data set that's been continually improved, and in terms of the amount and type of information that's available and the accuracy of the mapping itself. And so that was the foundation of this project. And so I guess I'll just close with the models and modules, and that's just the correlations that we found between these data sets that were incorporated into our model. I think that's a very important component, just understanding the power of the data sets. And then also because, we have a model, but the model is really comprised of a series of modules. The module is something that you can pull out and plug into the model based on project needs. So that means that our flycatcher salt cedar habitat restoration siting model is very easily adapted to pretty much any other habitat restoration or just straight up restoration siting. For the Middle Rio Grande, all we have to do is pull out certain modules and plug in other modules to get the results or to meet the goals that we need for any restoration siting circumstance in the Middle Rio Grande.
Okay, thanks, Andrea and Joe. If anyone does have any questions, you can put them in the chat box. That was a great follow-up presentation to Kristen's. I do have one question. Joe kind of talked about this, but with the yellow-billed cuckoo creating some challenges, obviously, so the strategy was just to avoid those sites, or could you elaborate on that a little bit more?
Yeah, absolutely. So this is Joe, and our approach was primarily to identify those sites, and yeah, if the habitat or potential flycatcher restoration site was located in close proximity to those sites, then we coordinated with the U.S. Fish and Wildlife Service to determine on a site-by-site basis whether or not it was feasible to recommend those sites for flycatcher restoration. And so really we were just asking for permission and getting guidance directly from Fish and Wildlife Service to get the rocks out of the road at the sites that we felt we could potentially still develop flycatcher habitat for.
Great, thank you. And so is there the possibility then of going back to the other semi-final sites that you identified, like future projects, if you could talk about that and the ease of maybe going into doing future projects now that you've identified that many sites?
Yeah, so we have a master matrix for all of the sites that we identified in the project. And so there would be 3 different tiers. The first tier would be the 216 sites, which we've compiled all the spatial information and analysis into a single matrix for reference. And then we also collected an elevated level of detail for information on the second tier semi-final sites. And that was also provided. That's the 50 semi-final sites, and those are also provided as part of the submittal for the project. And then again, for the final 9 sites, we naturally collected as much information, including field validation data, as we could.
And I'll add that, Reclamation has been looking at that information as well, and certain reaches that they're looking at doing projects. So this gives them a place to start, and then take that, in further detail depending on, what project area they're trying to work within. So a lot of that initial planning information is already there.
Yeah, that's great. It seems like you created a really nice roadmap for people to utilize in the future.
Yeah.
Great. Well, thank you both for your time. That's a great presentation.
Thank you all.
Okay, our last presentation is by James Hatton with the U.S. Geological Survey. The presentation titled Developing Southwestern Willow Flycatcher Habitat and Ranking Metrics at Multiple Scales with a Satellite Model.
Hi, I'm Jim Hatton, and I'm going to be talking to you today about developing a stream reach ranking system for the Southwestern Willow Flycatcher. My co-authors are Matthew Johnson and Jennifer Holmes, both of which have spent their careers out surveying for birds like the willow flycatcher. I work for the Columbia River Research Lab, the U.S. Geological Survey, and I have been involved with modeling flycatcher habitat for about 20 years now. So what we want to do here is to scale up from the original satellite model that I developed 20 years ago with Charles Pradzik in the Arizona Game and Fish Department. This is a 30-meter pixel model. It's very good. It's used all over the range of the flycatcher, and it's still considered to be very useful. But we also thought it would be useful if we could rank habitat at larger than the patch scale, which currently the satellite model uses the patch scale, about 120-meter radius. What we wanted to do is see if we could use the satellite model and extrapolate it over broad areas and then characterize characteristics of the riparian vegetation at that much larger scale, and then be able to rank the reaches so that managers could look at it and think about restoration and enhancement activities. So our methodology, we're going to run the satellite model over the entire Gila River basin, and we're going to characterize habitat in areas that the Fish and Wildlife Service delineated as flycatcher critical habitat. And there's 7 large reaches in the Gila River basin that are flycatcher critical habitat. And then we're going to compare it to the other reaches in the basin that are not flycatcher critical habitat. So we're going to use the flycatcher critical habitat reaches as training grounds for developing new habitat metrics that we will then apply in a rank model in order to see how many of those 7 original reaches we can correctly classify, as well as the remaining reaches in the basin. So as you know, the Gila River Basin is divided kind of right in two between Arizona and New Mexico, and it has quite a few flycatcher breeding grounds on it. So we thought that it would be a good location to try out this new reach rank model. And if it works well here, then we thought we could extrapolate it throughout the flycatcher range. So what we did is we divided all the reaches of the Gila into what we call zones. So we have the 7 critical flycatcher habitat reaches, which you could see are the red polygons. And we also delineated polygons for the other reaches. So there's a total of 60 reaches that we identified. Each one is a zone, meaning that a polygon's been drawn around the reach. And that enables us to use GIS zonal statistics in order to characterize the flycatcher habitat. It's an extremely efficient way to model large areas and large data sets. Quick overview of the original satellite model. It's 30-meter resolution. It was developed along the Gila and San Pedro and at Roosevelt Lake at the end of the 1990s. I helped develop that model as the lead modeler, and I worked with the flycatcher surveys for the non-game branch of Arizona Game and Fish to develop the model and test it. And what it does is it relies on satellite data. It uses the normalized difference vegetation index in order to identify the density and lushness of riparian vegetation in 30-meter cells. And then we segment that using different scripts to look at how much vegetation falls within a 120-meter radius of any cell. And then we calculate this. We can literally calculate the size of the patches, and we can calculate the edges of the patches and the heterogeneity in that vegetation. The model's been tested quite a few times. The last big test was in 2016 when I extrapolated the model range-wide and actually looked at the density of flycatchers within the 5 model probability classes. And as you can see, it has an exponential relationship. So you're about 30 times as likely to find a flycatcher in a 5th probability class as you are in a 1st probability class. All right, so how did we do it? We ran the satellite model and created 4 new variables that characterize the habitat in 60 reach zones. The first variable was how much habitat greater than 40% probability that the satellite model calculates is in each reach zone. So it's actually calculating the percent of the reach that's covered in habitat. We also looked at the top 3 model probabilities, classes 3, 4, and 5, and we averaged those. So what we're doing there is we're trying to discern whether the highest probability classes, are a good delineator of habitat. We also looked at the average number of years that flycatcher habitat persisted between 2013 and 2020. So we ran the satellite model between 2013 and 2020, about the same date every summer, and then we averaged that, summed it up to see which cells had the highest values and which the least. And that corresponds to the number of years in which habitat persisted at any given area within that reach. And then lastly, we used the NHD plus GIS layer, which is the National Hydrography data set, to assign the largest annual flow to the reach. So NHD exists for the whole country. It's a beautiful data set. We are able to use zonal statistics to extract out that maximum annual flow and assign it to each reach. So this is what it looks like. On the left now, we have a map of model probabilities of greater than 40% averaged across reach zones in both the critical and the non-critical flycatcher habitat. So this is one of our four predictor variables. And then on the right, you can see we just use the zonal statistics to average those values across the zones. So the orange group is your critical flycatcher habitat, those 7 reaches that you can see delineated. They have, we compare the values that we found in those reaches compared to all the remaining reaches out there. And you can see there's a very large difference, much more area covered in predicted flycatcher habitat in the control group. The second model was the top 10 probability classes averaged across the reach. And once again, you can see that there was a clear delineation. Those 7 reaches, if you want to think of them as controls, they had a much greater, much higher model probabilities when averaged across those reaches than the remaining 53 reaches. And then the third plant variable, all derived from the normalized difference vegetation index, persistence. So what we did was measure persistence across those 60 reaches and within those 2 groups, critical flycatcher habitat and non-critical. And you can see that predicted habitat persisted for much longer periods in the control group than the non-control group. That's the first time that I'm aware of that type of analysis has been done. And then lastly, we looked at the average annual flow in each zone. And you can see that for the critical flycatcher reaches, there was much higher average annual flows than in the non-critical reaches. Okay, so now I'm going to show you how we rank the habitat. Those box plots that we looked at, we can derive the 25, 50, and 75th percentiles and use those as probability thresholds to create binary predictor maps, zeros or ones, for each of those 4 variables that we looked at. So we experimented, looked at the 25th probability percentile and the 50th percentile, and we decided the 25th percentile did a better job at delineating those 7 critical habitat reaches. So from here on out, I'm talking about the 25th percentile in that control group to create those binary habitat predictor grids. And here they are. You can see the 4 that are used now for the ranking scheme. Predicted habitat, 25th percentile on the left. The top 3 probability classes, lower right. The flow magnitude on the lower left, and then habitat persistence on the upper right. So these are zeros and ones now. Anything above the 25th percentile of that control group we use to create that binary grid. And then we averaged them all together. We stacked them all together. So now you have cells with values from 0 to 4. If a cell had a value of 0, that meant that none of the cells in that reach contain one of those 4 predictor variables above the 25th percentile as compared to the control group. And if it had a value of 4, it means that all 4 variables said, yeah, you're above that 25th percentile. So that's the highest probability class. All right, so looking at this ranking system, the 60 reaches, you can see that there's 2 important breeding grounds that we just wanted to quickly check. One is the Gila Cliffs on the right, and the left is Fort Thomas on the left. They both have a lot of flycatcher breeding. And we just wanted to see if this ranking scheme did a good job of capturing those areas. And it did, so that gives us some confidence that this methodology is working. Of the 60 reaches that were ranked, 30 had 0 values, and we don't think they have much conservation potential for the flycatcher. Of those remaining, they just broke out that some were found in class 1. You can see some main stem habitat, which surprised us. Others, class 2 and class 3, are found in tributaries and a little bit in the main stem as well. And then the 4th highest rank is all were found in the main stem itself. So it appears that it's sorting these areas out by hydrology and by the characteristics that we identified for the flycatchers. Okay, the methodology appears useful and repeatable. We really think that this is a methodology that has great potential to be extrapolated through the range, as well as refined regionally, because you can apply the model to any area, and then you can actually modify those thresholds for those 4 predictors. So if you have a control group, you can always apply the same methodology, look at those box plots, and look at which percentiles do the best job of identifying your control reaches. And then you can literally use that set of values to run the model in that a different area. Okay, so we think that the model appears to do a good job of pulling out the poor habitat reaches and areas with low persistence of habitat. And we want to emphasize so that that's not necessarily bad for that predictor, because flycatchers like flooding, they like heterogeneity. So floods can really knock down the older trees, and you can get young willow and sprouting up, and within 4 or 5 years, those flycatchers are all over those areas. But it can also identify the areas that we think have been impacted by fires, insects, and droughts. So we feel good about that. The model can be used to determine what factors are limiting. So remember, we have 4 predictors, so we could do an analysis for any given area and identify whether it's limiting by hydrology or it's limited by the persistence in habitat. And when you think about it, that's super handy for managers to know. We do think the model needs to have further testing and refinement in order for it to be as useful as it might be regionally. But I've already identified or said that it's so flexible that we think that we could extrapolate this to other areas and do quite well with it. We do think that additional development of the model at finer spatial scales could be used to guide restoration enhancement actions at the subreach, patch, site level. So here we use reaches that were kilometers long, many kilometers long. But there's no reason why you can't use this approach on 100-meter long reaches and use the same approach. And I think it would work well. So that's what I have for you. I hope you've enjoyed the presentation. I'm looking forward to answering any questions that you might have. Thank you.
Thank you, Tim. Great presentation. If anyone does have any questions, you can put them in the chat box. Just a few comments for you, Jim. I think that it's really exciting that this methodology is repeatable, and particularly that it really identifies areas impacted by fires and drought. I think that's really on everyone's mind, right, with the increased fires we're seeing across the Southwest.
Yeah, I do think it's an extremely flexible modeling approach that we could divide the river reaches up into 100-meter sections and run this same model and just get a much finer scale. But it's also useful to have that coarse scale. A lot of times I think there's different issues and questions that you're looking at. So it's great to have a model that can work in both directions. Yeah,
absolutely. Do you have any interest or desire to continue on with doing that at a finer scale in the future?
Yes. I mean, we were just really testing this as a concept, and the more I worked with it, the more I liked it. I mean, it's what you saw, this presentation was really a thought experiment. Those reaches are so big, obviously, that people say, hey, wait a minute, there's flycatchers here, they're not here, they're not there. But the very approach is so useful at a finer scale that you could just turn around and segment that river and run it at that finer scale and then have 2 different scales of analysis. the habitat flycatcher software that I put together, the Swiffle Viewer, that has the predictor variables 2013 to 20 in both the top three classes and the probability greater than 40%. So those three plant predictor variables can all be created from the output of that Swiffle Habitat Viewer. And then all you need is the NHD plus data set to get that annual water flow, and boom, this approach is portable for anyone who wants to use it.
That's great. Thank you for that. On a lighter note, maybe to end a very long day, someone wants to know if you're working in your sauna.
No, but it's almost as good. I'm in a little cabin in the middle of nowhere by a wildlife refuge. That's a true story.
That sounds lovely. Let's see, Jim, will you share your riparian habitat areas, GIS, once you are done with your sauna?
Yeah, I mean, like I say, I could put this on the Swiffle Viewer that I've created and made available to everybody. And also maybe take a first cut at using this approach on a much larger area even, or in different areas, and then put those out there maybe as a way to get the discussion going, and then maybe a follow-up workshop for people. Because I really think this methodology, once people realize how flexible it is, I really think that people would like to use it.
Yeah, absolutely. I think it's a great idea. Yeah, follow up to that. Pamela says she'd like to use consistently digitized or vector areas to drive greenness and ET.
That's perfect for that. I mean, you've got the grids. Well, actually, I put them out as shape files on the ArcGIS Habitat Viewer, so they're all ready to go. You can download those 2013 to 20 predictor variables. They're already in that format, vectorized, I guess you could say. So there's a lot of flexibility here. Maybe a follow-up workshop would be very useful.
Yeah, I agree. And yeah, request to send the link, which we can connect people to all the resources that you have gone over in your presentation. So
yeah, thank you.
Yes. Are you able to put the link in the chat, Jim?
To the Swiffle model? Is that what you're referring to?
Mm-hmm.
Well, sure I can. Let's see if I can do it here. Hang on a second.
Looks like someone's...
Just 1 second, and I'll come back.
Oh,
Hopefully
so Pamela, she did say to where to find the riparian boundary files.
Oh, good. Okay. And here is the link. Yeah, there it is. That's great. To the Swiffle Habitat Viewer. So you get 2013 through 20 on that for both the binary and for the top three probability classes. Persistence, just add up those last 8 years, 13 to 20, add them up in GIS and divide by 8. And you got your persistence variables. So it's totally doable.
Great. All right, Jim, thank you very much for presenting, for your time. I really appreciate it. Yeah, you're welcome. Okay, that officially ends our last session of the conference. So over the last, from last week and this week, the 4 days that we had over 100 attendees, or close to 100 attendees. So we can't thank you all enough for participating, for your flexibility. This has been a interesting endeavor for us to do it over Zoom, but I think it went well, and we're all very happy with the outcome. So thank you all. Thank you to all of our presenters. We obviously couldn't do this without you. Another shout out to our sponsors. So thank you for supporting ongoing education and continuing the conference even in the time of COVID. So a few other reminders: if anyone does want to stay on and continue the conversation, we can create breakout rooms for you. Just put it in the chat. We'll leave Zoom open as long as you'd like. And tomorrow, again at 9 a.m., we'll start our workshops. So thank you all. And if you have any other comments or questions, feel free to put them in the chat. Oh, and one other thing, if you are not attending tomorrow's sessions, if you could, please fill out the conference eval. We really take that seriously to try to improve the conference every year. So thank you all.